Hello and welcome everyone. My name is Carmen Guguta and I'm your host today from Technovis Crystallization Systems. Together with Edwin Arret from Mercachem Syncom, we are bringing to you today the webinar on optimal form screening and selection by controlled crystallization. This webinar is being recorded and will be made available to all of you. Most probably tomorrow you will receive an email with the link to the recording. This evening I'm sure you will already be able to watch the recording on our website crystallizationsystems.com or on our YouTube channel together with all our past webinars. You are being muted at this moment, but if you have any questions, please feel free to write to us by making use of the chat function of this webinar platform, or you can write to us in the coming days at info@crystallizationsystems.com. We will do our best to answer all your questions together with our presenter, Dr. Edwin Arret. This webinar will last about 45 minutes, including questions. I'm happy to see actually that quite some of you joining us today are already users of Crystal 16 or the crystalline instruments, but for those that do not know yet, Technovis Crystallization Systems, we are a Dutch company located in the north of the Netherlands. I am proud to say that we are active worldwide since 15 years ago with more than 500 instruments sold globally. Our portfolio consists of 3 products for crystallization, process development and optimization, and formulation related research. I will just briefly introduce to you in 1 minute our instrument, and then we go to the presentation of Edwin. Our instruments, the Crystal Breeder, Crystal 16 and the Crystalline, they are multiple reactor systems that you can heat and cool in a controlled manner. They have a small footprint and with little training required. Software is user-friendly. You can use cheap disposable reactors, so you do not have to worry about cleaning the reactors or analytics. I'm actually proud to say that we are the only company on the market with inline analytics. We do not have probes that you have to place in your reaction vial. So you do not have to worry about cleaning those probes or cross-contamination or even changes, modifications of fluid dynamics. So you do not have to worry about any of those things. The Crystal Breeder works at microliter scale, the Crystal 16 at 1 milliliter scale, and the Crystalline at 5 milliliter scale. The Crystal Breeder is your instrument for discovery, for early stage type of crystallization screening or growing single crystals. As I like to call it, is the gadget type of instrument. It comes with a lot of gadgets. Any type of crystallization method is available on this instrument. Evaporation, controlled evaporation, vapor diffusion into solids, into liquids. It comes with a quite smart setup for that. Cooling evaporation, thermocycling, slurry, layering technique, you name it. Everything is possible on this instrument. On the other side, the Crystal 16 is your instrument for solubility determination, temperature-dependent solubility. In 4 hours, you can determine the solubility, the temperature-dependent solubility curve of your compound. And not only that, you have also information about the metastable zone width and the supersaturation curve. Any type of induction time measurements, nucleation rates, you name it, it is possible to be performed on this instrument. Polymorph, salt, co-crystallization type of screening, even amorphous solid dispersions you can perform on this instrument. Crystalline. Crystalline is the instrument, as I mentioned, works at 5 milliliter scale. Slightly larger than the other ones, and with the camera technology that works exactly like a microscope, you are seeing always what it happens in your vial. And as I mentioned, these cameras are your eyes on what happens in the vial from outside of the vial. It is an instrument that allows you with this technology to develop or optimize your crystallization process to control form, habit, particle size distribution, in formulation. If you have a Raman spectrometer in your lab, no matter the brand that you have, you can connect it to this crystalline instrument and follow chemical reactions and polymorphic transformations as usual. If you would like to learn more about what these instruments can do for you, we can set up an online demonstration, and nowadays it's not that easy to reach physically a lab and not safe as well. So if you want to learn more what these instruments can do for you, please ask us for an online demo. We can set up a session depending on your preferences. And now I'm really happy to introduce to you our speaker today, Dr. Edwin Arrett. Edwin and I, we are going back to, we were just talking back to 2004, when I just started my PhD at Radboud University Nijmegen. And when we met later on at Avantium, working together, we even shared an office together. Thank you very much, Edwin, for accepting to present this webinar for us, and I'm really happy to be able to work again with you, at least just for a short while. For all of you, Edwin holds a PhD in solid state chemistry from Radboud University Nijmegen in the Netherlands, a beautiful city in the Netherlands. He has nearly 25 years of crystallization experience, supporting every facet of the pharmaceutical development cycle from discovery to commercialization. Edwin's in-depth expertise includes high-throughput screening crystallization, form selection, stability studies, solubility determination, and crystal habit optimization. Edwin is currently the principal scientist for solid-state chemistry at Mercalchem Syncom, which is nowadays, maybe you heard, Cymeris, and he is leading his team of 8 solid-state chemists. And now I give the floor to you, Edwin. Let me make you a presenter. I think now you should be able to share your screen. Once again, thank you for doing this, Edwin. Yes, we see your screen.
Okay, thank you, Carmen. Yeah, we go back quite some time already, and indeed, we show now what has been happening for the past, well, almost 25 years that I'm working now on actually crystallizations and everything that is related to form selection. Since a few days, we have a new name. All the sites have the same Cymeris name at this moment. We are located in Weert in the south of the Netherlands. And I will present you looking at form screening and selection and focus on controlled crystallization. What can you learn from the crystallization? What can you learn from looking at the process itself? A short introduction on solid state chemistry. What do we do here? We are supporting the pharmaceutical industry. We don't make any drugs ourselves, so we only support the first stages from discovery up to the clinical phase 2A. Which means we also have the first GMP materials. We start with crystallization. Actually, we look at Viscam properties. We have the solid form screening and selection. But next to that, we also need a process. So how to make that form, which you select from your screening. So we support the process R&D. We work on pre-formulation to make sure that the drug will end up where it needs to do the work. And we focus also on optimizing the crystal habit. In this presentation, we talk about crystallization and solid form screening. What to do when? Crystallization is in the whole process of research and development, and actually when you produce, you also want to check if you really have the same form each time. So in discovery, we look at the first crystallization. You want to have a single crystal, so you know the structure of your material, and you're looking at solubilities. This kind of characterization program lasts about 1 week of development needed. Once you go to preclinical, you want to have a selection of your form. Either it is a salt, a polymorph, a co-crystal, or you work with amorphous. At that stage, you're also going to look at the crystal habit. What kind of material do you have? By producing the material at somewhat larger scale, you will see what the isolation, so your filtration and drying properties are, and if you need to optimize that part as well. The solubility goes into pH dependent solubilities or simulated fluids, and the start of the process development is here. Typical program 1 to 2 months. Finally, in phase 1-2, you can have the IP screen. You produce a GMP material which needs to be optimized. DOE studies on that one. I will not talk about that part in this presentation. We look at physchem properties. So, and the main target there is to balance these properties. So if you look at a typical classification of drugs based on your solubility and permeability, most of the marketed ones are now in what we call the class 1 with the high permeability and the high solubility, which actually gives no problem to put on the market, and they will do the work. The ones that we currently get most are the ones with the low solubility, but they do have sufficient permeability. So there are about 30% is marketed, but the candidates that are put on the market is 60 to 70% of every compound that we get. And the main question there is, we have a low solubility, can you improve that? And of course, if you want to have the most stable form, that one will also have the lowest solubility. So that is something that is related to each other, and we can have a look how to optimize that part. The ones that give high solubility but low permeability, the candidates are very few, and when they're marketed, it's okay to use them. It becomes an issue when you have low solubility and low permeability. A few are only marketed. We have quite some candidates in that field, and most of them will not make it to the market because of their properties. So looking at physchem properties, it's a balance. It requires your solubility, the stability and permeability of the compound. And actually looking at the physchem properties will put your compound somewhere in this field. It will give you a certain development strategy, and it is also a certain risk assessment. What risk are you willing to take with your compound? The aim is to balance all your properties and give you a blueprint how to develop it. Looking at your solubility, you can change your polymorph, you can look at hydrate formation, you can change your particle size. If that doesn't work sufficiently, a salt or co-crystal might give you the purpose. Looking at permeability, you cannot do that much on screening. So you have to change something on your molecule, which means you can have a look at a prodrug, you can have a complexation or carrier. In stability, of course, your crystallinity, your quality of your crystal will give you a higher stability, but you also need to take into account that your material might be hygroscopic and you want to avoid that part. When going to crystallization, it's all about control. So if you ask what is crystallization, you can get a whole list of different answers depending on who to ask. So it can be self-assembly, it can be just a separation technique. I want to have a solid. It can be a purification method. You can have a certain property, look at your crystallinity, look at polymorphism. And it is also IP protection of the molecule. it's getting more value to your compound. By looking at how does amorphous look like and how does crystalline look like. This picture says it all, and actually the control of your crystallization will allow you to give you the crystals to the right. And the whole idea of the process, that what we do, is to go from the amorphous solidified oil to a crystallized product. So crystallization. Of course, you can measure a lot and you can put a lot of theory behind it, but the idea is, if you want to control something about your nucleation, it is driven by supersaturation. That is the reason why it starts to nucleate. And that is actually controlled by your concentration, actually your concentration difference. So what is your actual concentration compared to the equilibrium concentration at that condition? If you steer that one, if you have a higher supersaturation, it will nucleate in a different way or faster. When you look at crystal growth, most of them are, let's say, depending on what kind of solvent do you use. How does it want to grow? Does it want to come out of your solution, and does it form a nice stable crystal? Of course, you can steer it in a direction where you have control of the mixing. The third one, and most of the time it is being neglected actually, it's your isolation. Your filtration and drying of the product will certainly have an effect on the quality of the crystallization. So sometimes a filtration is actually the factor that is depending on which form to select and which crystal habit to select. Next to this part, there is also what is actually kinetically accessible. So which forms can you make? Which form is long enough stable to identify? And that can be reasons for a patent. It can be reasons to get a certain form available. Which form is thermodynamically most stable? So there is one single form in which all others will convert into, but what are the conditions required to get to that most stable form? And what is then pharmaceutically acceptable? Solvates are not. So you have to work with anhydrates or hydrates. And there is a limitation in your selection of the counterions and coformers, depending on what formulation you actually want to use. So knowing your formulation in the stage where you have a selection of which form to take is already a pro that you can get to the right form and the right conditions. Going to solid form screening, I can say it's never enough. It's never enough time, never enough experiments, and never enough material. So when you are in discovery phase, it's actually when the first milligrams of material are isolated and you work from a chromatographic isolation to a crystallization. What we do is a thermal cycle in 16 different commonly used process solvents, and we avoid complete evaporation. That will give you a solid, but not a good crystal. We avoid freeze drying and spray drying. That one will give you amorphous compounds. Yes, it will give you products, but not the crystallization that you're looking for. In this case, the solvent selection is based on actually the class. So what is being used by the chemist to get in the process to get this material? The ICH class, or is it commonly accepted, is not leading here. Just get a solvent that works. And we aim for single crystal growth. It will confirm what is actually your molecular structure there, but also it will allow you the first patent on the molecule. There are many different crystallization methods, and this is just a list, an overview, and it shows that when you're working in research, you have actually the mild conditions close to equilibrium. But when you go to production, when you want to make sufficient grams or kilograms of your material, these are absolutely non-equilibrium. There's not enough time, and the conditions are such that you actually want to have high quality, high yield, and good purity of the material. So that will give you only a limited amount of options of the methods that you can use. In discovery, whatever method works. You want to have that first crystallization, so you're not bound to actually what is a good way to scale up, what is a safe solvent to use. You just want to have it crystalline. In preclinical, only use the scalable methods. And a few are mentioned here, and not all of them are that scalable, but at least if you cannot have anything else, use these. In the crystallization methods, you work with the temperature-dependent solubility. So you work with a cooling crystallization. Your solvent selection is the solubility curve, which one is fixed. The nucleation curve is the one that you can change, and that depends on the conditions that you have, your crystallization conditions. So you can still have different forms from the same condition, your same solvent, but you can also have different qualities. And that is something that you don't want. You want to have a consistent form, consistent quality, and each time a reliable result. So that requires a lot of experiments to do. What kind of equipment can we use? Well, of course, working with crystallization requires quite some parallel experiments. So you don't know which one will give you the right result. So there is the possibility to work at 3 different scales here in parallel, depending on what actually is your solubility. That's the first one. So if it is very poor soluble, you first go to your crystalline where you have at least 8 milliliters of solvent present, and you can use that one to get to your first crystals. If you can find a very good solvent, the crystal breeders will be sufficient. If you want to make anything amorphous just to have material out, you can still use your crystal breeder there. Why should you use this kind of equipment? It's the only one available that gives you stirring, heating, cooling, and turbidity with all in one equipment without actually disturbing your crystallization process. You can follow it, you can look at it. Some your crystalline has the video in it, so you can still have a good overview of what is there. So knowing your crystallization, understanding your crystallization, that will give you a good product. Once you have your first crystalline material, the scale-up steps is about 5 times the volume, which also gives you a good following from crystal breeder to crystal 16 to crystalline, and then it goes to liter scale actually. In the preclinical phase, you start when actually a suitable process is known, so you can make at least a few grams of material. You can have sufficient material of good quality. You look at scalable methods, which focus around cooling crystallization and anti-solvent addition. You look at a solvent selection, which is based on diversity. So principal component analysis based. That's one that I come back in a later slide on. These solvent selection is pharmaceutically acceptability is leading here. So here is your ICH class is leading. Can you actually get an anhydrous form with the low limit of the residual solvent in there? You need to find the kinetically accessible polymorphs. So which forms can you make and which forms are stable long enough to detect actually? Determine the thermodynamically most stable form, and most of the time this is also the form that you will take, but also know the conditions at which it is the most stable form. That is very important as it is part of your selection process. And find in the end the crystallization which gives you the highest purity and yield. Going through these steps will show which form to take actually. Very short on polymorphs, just a reminder, same molecule but a different crystal structure, and the main important thing is that the properties differ. So your activity as a drug will differ, all your thermodynamically as well as solubility as stability will be different from it. So you need to select the one that you can make, that is accessible, that you can have stable long enough under normal ambient conditions. But also it provides you a patent again. So each new polymorph is a novel substance which you can patent. When we go to solid form screening, there are 2 steps in it. We use high throughput screening that will give you the broad overview. A lot of experiments, a few hundred, but it gives you very limited amount of information on each of these experiments. We use automation to avoid manual repetitive handling. And it's the basis on what do we actually have, what is the landscape of the crystallization behavior. Next, we use the single experiments for more detailed. We need to mimic actually the reactor conditions, at which scale, at larger scale, you will make it. So you need to have the top steering. You need to have a good control of your crystallization, of your temperature, of when does it start to crystallize? Do we see anything that is changing in time? Do we get another form there? So making the screen work will depend on actually which solvent to select. So if you look at the structure, we have about 20 classes that you can use, but only selecting based on that one will not give you a good diversity. So what we have done is define which principal component is most important for this part of the crystallization process. So polarity, polarizability, and hydrogen bonds. These are the 3 main properties of a solvent. And when you combine these 3, you can put it with a calculation in a 3D space here. You want to select the solvents which are as far away from the center point as possible in each direction. So the red dots in this part. You can select 8 different ones and the center point actually. And that will give you a good feeling on how does it work in the total solvent space. So you can have prediction how does it behave, but you will also be able to select similar solvents which will do a similar behavior there. For overview screening, you want to have highest diversity, but for IP screening, you want to have the most different solvents, so the number of solvents. Again, a different approach there. When you look at high throughput screening, there is also high throughput analytics required. And that one, although X-ray is widely spread on the market, high throughput analytics that does the trick of actually analyze a full screen within 24 hours, the time frame that we also need to do the screen itself. That one was not available in a format where you have a high resolution and a high throughput. So we built together with the Bruker this kind of equipment, and it is on the market right now, which allow you to give with a very limited manual handling, no sample treatment, give you a good overview of what is there within 24 hours and only giving one order to do the measurement. Of course, the solution that you have, that is readily available, so you can have your LC measured, but we added to the program that you have automated dilutions at 2 temperatures where you can have a look at the purity as well as the solution stability of your material. Often I get a question, but which polymorph do I need to select? Because then I have, let's say, 400 experiments done, and you got a lot of results, and you get an overview like this, and you have no idea which one to select. For sure, the most occurring one is not the one that is necessarily the most stable one. And your design of your study determines the outcome. So if you have a limited amount of different solvents, if you only have a certain amount of starting material that is already the most stable form, and your conditions, that will give you already the outcome of your design. When you look at what is available, so you look at the Polymorph study, this is what you can find pretty easy on the internet, how to do a certain screen. It will give you an answer. It will give you not the overview, because this is a very rigid, limited amount of actually experiments, diversity of your experiments. So when we look at it, it is start with different concentrations, because that will allow you the difference in your supersaturation and therefore your nucleation behavior. When you do filtration, look at different temperatures. A slurry plate, start with the least stable one. So see if you can find an amorphous form. Also work with different temperatures there. Cooling, work with different cooling rates. It will give you different results, of course. Anti-solvent, there is a lot to be very in that one. You have forward and reversed. You also have your anti-solvent in evaporation into a solution or onto a solid. Vapor diffusion instead of evaporation, so a controlled atmosphere or an exchange of your organic solvent by a more humid atmosphere. That can also give you different forms. And also on your HPLC, look at different temperatures. That will give you an idea on actually, does it give you a process that allows you a cooling crystallization? Is the difference in the high temperature and low temperature sufficient? A lot of information you can get already from this study. But the study itself is not giving you the optimal form. It gives you an overview, and your answer is somewhere in there. So the next steps are, look at your material. Use TGA, is it a solvate? If so, you cannot use it. Do you see what kind of thermal events are there? Look at your energy, look at the temperature at which it starts to melt. Do you see other events occurring? Recrystallization will give you immediately the idea, okay, this is not the most stable one. So learning already from a few milligrams of material. Reproduce the experiment at larger scale. That is why we have the different Technoviz equipment in-house. You can do a full characterization, you can follow actually what is ongoing there, and you have mutual stability. You can make your solubility curves. Here's an example of two different forms which we found in a screen, and it is actually depending on at which temperature does it allow you to crystallize. It is about 55 to 60 degrees, where's the transition temperature, which is a temperature which might occur in crystallization of your compound at high concentration in production, while in your lab you have a low concentration and it starts to crystallize at 50 or 40 degrees. So you come up with the wrong form, as it is a different stability there. You need to know conversion conditions. Reproduce that form at gram scale to see what do you have, and that is the point where you also have to look at your crystal habits. Do we have a powder, or do we have a sticky material that actually shows no flowability? To optimize, you need to look at your material. So form ranking is very important there. Stability of your material, competitive slurries, but also look at your melting point and what is the melting behavior. Is it a true melt, or is it a loss of a residual solvent which is ongoing there? So it needs to be producible. That is the aim of the studies that we do here. Make one form that you can produce. All the other ones that give you more information, and you can select that analytic that is giving you that kind of answer that is required. When you have the different forms, know how to convert one form into the other. So these are the ones, the ranking, how to conversion of one into the other, and that will allow you to choose the right one, the conditions that you need to avoid, but also the conditions that you need to see, can they occur in stability studies, in storage conditions, in sample handling. Questions I get as well is, okay, how to make the selection of the best form? And here I got an example in which different companies had a look at a certain material. According to a solid state company, this crystallization of form 1 was excellent. You get a nice free-flowing powder, and the properties of that one for a solid state person were excellent. Looking at how to make your material, so from a process chemist, no, absolutely not. We have these kind of nice, well, large crystals, filtration rate excellent, drying times very short, and you have an optimal form here. A totally different form compared to what the solid state actually has as optimal form. Finally, it ends up in a formulation, and it shows it forms these very long needles, which was a solvate, a hydrated form. That is the result. So by starting from just a free-flowing powder, we end up with a very viscous, well, actually, you cannot use this kind of formulation. So if we knew what the formulation was in the part where we started to look at the material, when we had the solid state going on, then we could have steered it in a certain direction, but also avoid your solvate or your hydrate formation there. It could give you a good process and support that process, optimize your particle size in such a way that it is an acceptable filtration and drying, and give you a good formulation instead of this. Well, you cannot use this one. Finally, when you have the question on why should you look at your metastable zone width, so why should you look at your solubility curve and on your nucleation curve? It's quickly measured, that's one. So it's not that it's kind of an automated way. You prepare your sample and it's being measured. So it's easy to do. It gives you, in this case, a good solubility curve, but actually it shows already a poor nucleation control. So it arises certain questions in the phase where you have experiments at 1 milliliter scale already. So should you use another solvent or solvent mixture? Is your cooling rate okay, or should you lower the cooling rate and get a more control on your nucleation? Maybe if a cooling crystallization doesn't give you really that good result, should you use an anti-solvent to start your nucleation? Or what is the good range to have your seeding if you want to have complete control of your process? On the other hand, when you look at a cooling process, you can also see down to what temperature you should go. Going to below 0 temperatures are costly at the larger scale, easily done in a lab scale, but if it doesn't give you more yield, and that shows in your solubility curve when it flattens, then that is the limit. So the technical loss that you will have on your yield. It gives you an idea how to put your process and where should you stop in optimizing that part. Looking at your form screening and form selection, it is always different than you think when you start. So it's continuous troubleshooting. It is giving you a good broad overview, and each time your conditions will tell you where is your optimal, and each time it will be a different optimal when you have a different compound. Understanding your crystallization behavior, that will manage your risk and avoid anything that you don't want to see, any changes that you don't want to have in your process. When you have your data available, accessible and searchable for a whole project team, so not only for the solid state chemist, but also for the organic chemist, as well as for formulation, that will give you a good success. Because if you have a report with only those experiments that worked and not including the ones that failed, that will give not any overview. It will only direct you in a certain optimization from that part of your research. using automation, parallel experiments, that will give you a good overview. It's fast, you have control on which conditions you use, you can screen for different factors and have a good diversity there that will give you an optimal result. And if there are any questions.
Yes, thank you, Edwin. We have a few questions. But just before I start reading to you the questions, I have myself one question, or maybe two, related to the Crystal 16 and the crystalline instruments. You are a user of these instruments for quite a lot of time now. What would you advise? What to the ones that they listen to us today? What are the main applications for which you are using this, the Crystal 16, and then also the crystalline? How would you advise our attendees to use these two instruments?
Well, it's actually it's control of your crystallization. So when you do the parallel experiments, you vary that part that you want to vary, so a solvent or concentration or anything like that, and not cooling rate or sample handling or wait for the next sample to do. So parallel screening is very important in this part to make a selection. When you have a choice to be made, you can just put there the same condition, the same salt or the same co-crystal, and see how does it behave in time. You can follow it without disturbing actually the process of crystallization, and therefore also you have a good idea on is there any change. So we have a consistent form each time. You can just do 4 or more experiments at the same time, the same conditions. Do we get 4 times the same result there? That gives you more certainty on that one. Crystal 16 is, let's say, medium throughput screening. So it will give you a lot of information on the crystallization itself. When you go to the crystalline, it adds actually the crystal habit. So what kind of shape and size of the crystals do you get without actually grinding your material with a magnetic stirrer or anything. So from that scale, you have an idea on how does it look like, as well as something that you can use for the larger scale. So the results you obtain there, you can follow in time. Is there any change? And you can use it when you go to 50 milliliters up to liter, up to 20 liter scale.
Perfect. Thank you, Edwin. Now, we have a question from William. How much material is required to make a broad crystallization screen?
Okay. Of course, it's depending on how soluble your material is. For a crystallization screen, using the crystal breeder, we can work with about 100 to 200 milligrams already. So that is not the broad screen you likely will asking for. When you have the high throughput screening that we do here, it is about 3 to 5 grams of material, which gives you the 500 experiments and the total overview of which forms are there.
And somebody else was asking, related to the same question, but more towards the discovery phase or the early discovery. Usually, how much amount of material is typically needed? Of course, depending on the compound, the solubility of the compound, but minimum, what do you need for early discovery?
Okay, we work close together with our discovery site in Groningen, and what we get is sometimes even 50 milligrams up to 100 milligrams. So really isolation by chromatography and then evaporate to dryness. That is the material that we can get. On the analytics, if you can see it, we can make an X-ray out of it. So that is not really the part which we worry about. It is, do we have sufficient material and in a small enough volume, which means either go to Crystal Breeder or Crystal 16. The choice of the equipment determines how successful you are in recovering your material.
Okay. Another question, is the transition temperature solvent dependent? I guess transition temperature from one polymorph to another one or so. Is temperature is solvent dependent?
The temperature point itself is not, but how easy it goes from one form into the other, that is. So your conditions will give you how easy do we get the other form. It will always be a range in which either both forms can occur next to each other, or one form is a little bit more stable because the conversion, so actually your energy barrier that you need to cross is a bit higher.
Okay. We have another question. Under what circumstances, if any, would the speaker recommend selecting a kinetic anhydrate over and above a thermodynamic anhydrate as commercial available form?
Yeah, okay. First, when you select an amorphous one above a crystalline one, you need to stabilize that one. So you cannot just go for it and say, okay, this is the form to go, and we hope it will be stable enough and long enough. So that is first. Second, if your hydrate is under ambient conditions more stable because of the hygroscopicity of the anhydrate, please go for the less stable hydrate, because that one gives a lot of problem if you need to have your conditions such that you actually have an anhydrate that you cannot handle under normal ambient conditions. These are the two exceptions. I know that was not the question. When you want to go for a kinetically accessible form, it depends on what is needed to go to your thermodynamically most stable form. Especially are these realistic conditions that might occur. So in time, yes, it will go into the most stable form. Of course, that is a risk. But if you choose the kinetically accessible form, that might give you a benefit on your solubility or your dissolution rate.
Yeah, thank you. And I would like to add that indeed, if you look at the generic pharma, they are mainly looking for also for those not really the most thermodynamically forms. So their door into the market is actually not the most thermodynamically stable form. So there are quite a lot of drugs out there in that form as well. Another one, great presentation, Edwin. How many hours or days would you consider sufficient stability for a kinetically accessible form in order to develop a crystallization?
Yeah. Okay, difficult to give just one answer there. What we do is have at least a 3-day stability as a powder, open powder, so not in a package or anything like that. That will give you a good idea on any difference there. If you have different forms and you want to select the one, you need to measure in time. So measure after 1 hour, after 1 day, after 3 days, to see if anything changes. Also, once you have reached one form, there is a possibility that that is not the most stable one. So always have 1 or 2 more time points after it ends into one crystalline form. I think when you do a competitive slurry, 1 week is sufficient if you have a somewhat higher temperature or higher humidity as well in there.
Yeah. Another question from Frederick. Hi, Frederick. Would you do anything specific to develop a method to separate the stereomers by crystallization?
Yeah, okay. That's we have experts on separation on that one as well. So we work together then with chemist specialists on that one. We did some tests on making different salts as not as end products but as separation method. So you can have coordination or salt formation that will give you a separation possibility there. It's all about stability of that one. If it converts into the other because of the conditions, then of course it won't work. But there are many ways how to separate them.
Okay. Another question. Can you elaborate also on, a bit on the same, elaborate also on resolvation of racemates? Are your crystallization screens set up for these too? And the same question for salt screens. Is it useful for in situ formation of salt?
Okay, so when you if you can avoid to make this kind of mixtures, then of course that part is in the chemistry itself. We can work together with them to optimize that part. If you produce the racemic, then of course you need to have a separation there, and our standard way of working is not focusing on that one. So we need to adjust and that we require a few experiments before doing this kind of screen. So how does it react? How does it behave actually? Can we do a good separation there? And most of the time we work with our Nijmegen colleagues together on that one. On let's say making a salt in situ, I think that was another question. That goes into the formulation part. So you need to know the properties of that salt. But if your formulation is just a solution, then of course you can make the salt there, and there is no reason to isolate it as a solid. So these are choices in formulation that we can make if your salt shows poor stability as a solid, but very good properties to dissolve or to remain stable.
Another question, any amorphous product has been taken for crystallization and what are the results? So I guess if you could make a crystalline compound out of an amorphous product, I think, right?
Yep. So going from a clear solution, it doesn't matter what you put in there. There's a filtration step in between, so most of the time you will get different forms from it. When you start to recrystallize in a slurry experiment, please start with amorphous. That is, let's say, the least stable, and it will quickly go into another form, a crystalline form, if it is available. So it's looking at really at accessible polymorphs, and hopefully they are long enough stable to identify what actually your form is. So the time point at which you start your analytics, so when you isolate your material, it's very important when it is aimed for a kinetically accessible form.
Patents for crystal forms 1, 2 are applicable for industrial production?
I'm sorry, I don't understand the question here.
Patents for crystal form 1 and 2. Did you had any patents? I didn't hear you.
Okay, we have. I have an example where we have the two different forms.
Forms 1 and 2, yeah.
Which 1 and 2, which are anisotropic related. So Of course, both can be prepared. It is just the conditions of which one form can be transferred into the other. So, but in this case, the energy was very close. You can see that on the solubility curves, which are almost overlapping there. So only in this case, I can say, yes, both were feasible to produce. It is just the temperature at which you start to crystallize was depending on what result you got. We didn't find conversion of the solid from one into the other, which means that storage of the less stable one at low temperature had to be in complete dry conditions.
The plates, the Kapton plates that you mentioned you were using for the X-ray powder diffraction, are the crystallization experiments done directly into the wells of these Kapton plates?
Yes. So they are chemically good stable, and they don't give you any different results than when you use either glass or any other material for that.
Okay, Frank is asking us this question. Edwin will come back to you with an email, Frank, if you hear us, with more details on this and how you can access such a plate. We have another question. I think this is our last question that I'm going to take because it's quite a long time now. From Gustavo. Hi, Gustavo. Good to see you here. Great presentation, Edwin. In many cases of polymorphism, having 1, 2, 3, or some single crystal structures can give you more insight into the interconversion of them, and of course the packing of the molecules. We have gained a lot of information by using single crystal X-ray diffraction approach on the polymorphs. Do you agree?
Yes, absolutely. So knowing your crystal structure, that will give you a lot of information if it is accessible from one form into the other. That can be, especially when you have, let's say, channel solvates that easily give your solvent and the one that are only very, let's say, limited, different from each other in energy, as well as when you know the structure, you can see how easy they convert into each other. I must say that most of the time, the ones that are candidates for further production are very different in crystal structure. So if there is a choice to make, one is, let's say, stacking the molecules in one direction, the other one is going perpendicular to each other. So that is really a total difference, and a lot of energy is required to go from one form into the other. Having the single crystal data available, that is something that is very useful. And yes, we have single crystal capacity here, as well as looking at structures by calculation of the powder diffractor.
Perfect. Once again, thank you very much, Edwin, for helping us with this webinar today and the questions. We have a few more questions. I will send them by email to you, and you will receive the answers in the coming days from Edwin. For those that they want to learn a bit more about our instruments and what these instruments can do for you and in the lab, as I mentioned to you, we have this possibility to come online to you and to show to you and share our experience and knowledge with you on the different instruments, and hopefully you can learn a bit more about how we can help your research succeed. Stay safe, everyone, and hope to see you again in January when we start, early January, our new series of webinars in 2021, hopefully in a more healthy year. Thank you once again. Have a good Thanksgiving if you are celebrating this Thursday. Goodbye, everyone.